So the AP Biology equations and formula sheet. I'm going to make an attempt to explain this to you as quickly as possible without having gone over the information for it yet, just to keep you from being intimidated by it, because this is what it looks like. It's two-sided. It has all these crazy statistical things here, some kind of crazy chi-squared, whatever that is, all kinds of variables, and this is crazy looking. It's really looks horrible, but this is not that bad. Okay, so I'm going to walk you through every single individual part of this to make you feel a little better about it, hopefully. The good news is that whenever you have to use this, usually it is not in a very complex manner. Generally, it's not, unless it's one of the easier parts of it, in which case you should not have a problem doing that. So the first section of this is a statistical analysis part, which is, in my opinion, the most confusing part, because it's not a biological idea. It's just like pure math. Just wait till you have to take statistics in college. It is definitely a journey. So starting with the mean equation, because this one looks terrible, but this is literally just taking an average of numbers. So I made an effort here to point out the different parts of it for you. So we've got this little X bar here, which is the symbol for mean, and mean and average are the same thing. Okay. And then this 2 is pointing to 1 over n. So all that is a very complicated way of not putting this stuff in the denominator and then putting in the numerator and then having that in the denominator. So all that means is that whatever the rest of the stuff you do gets divided by n. And n is the sample size. So if you have 5 different values that you're using in your analysis that you're trying to average up, then n is 5. This right here, this pointy E is the Greek letter epsilon, and it means the sum of things. So it's the sum of whatever comes after it. The N on top means that you that's how many times you're going to do it. And then the I on the bottom is saying it starts with 1, so or it's going to have, you're not doing anything crazy to these numbers. So I don't know, just ignore this bottom part because it makes it look confusing, and I use it for counting purposes. So this xi, the way that I do this, and I'm willing to bet if I work out any of these problems, that I would probably do it correctly. Xi would be x1, x2, x3, and x1 is your first value, x2 is your second value, x3 is your third value. And my example here is if the first value is 3, second value is 4, and third value is 5, just to make it super, super simple. So what you're doing here is you have this right here, which just means you're going to make a big fraction. So we'll just take this and put it in the numerator, and then take this and put it in the denominator. And then you sum up all of the individual values. So you'll add 3 plus 4 plus 5, and then divide it all by n. So it's basically as if you had parentheses around this, and then so you're timesing that sum by whatever is in front of that. So literally, this is a complicated way of saying add up all your values and then divide them by how many you have. If you were trying to find the average of 3, 4, and 5, you would add up 3, 4, and 5 and then divide it by 3 because there's 3 numbers. That's all you're doing with this. So don't let this notation make you crazy because it looks awful, but it's really, really not terrible. You are literally finding the average of a set of numbers. It's just the way that they show it in statistics, which is terrible and awful, and I apologize, but it's also not my fault. And math is totally not my thing. So I try to explain it in a way that that makes sense here. So again, this is just the average. So don't let this right here freak you out. All this means is that you're doing this, you're adding up however many values you have. So you're going to be doing this 3 times because you have 3 values here, or whatever. If you have 5 values, you do it 5 times. What comes after it is you do that individually. So like if this was x plus 1, then you would do 3 plus 1 plus 4 plus 1 plus 5 plus 1 and so on. So that's what that means. So you add all that up and then you divide it by the n. So it's just an average. Don't let that freak you out. Standard deviation, the way that the formula looks is actually less intimidating, but it's a stranger thing to have to explain. So standard deviation is denoted by that lowercase s, and it's used to tell, like, if you have this group of numbers, it's a measurement that says how spread out they are. So if you have a bunch of numbers and all of them have the value of 5 point something, except for somehow you have like one that's 6 point something, and you have 100 numbers or something. So most of them are right there within one number of each other, and then you've got one that's sort of out to the side. So we're going to say, okay, is that going to affect how much the numbers are like spread out? What you're going to find when you do this is that if you have a whole bunch of numbers that are really close to each other, the standard deviation is really low. So it finds the average, and then it does some kind of magical something or other that gives you a value that tells you whether or not it's really, really spread out. So when we're solving for this here, we have that sum thing again, right? Here we have that xi again, so we do that for each of the x's, so x1, x2, x3, and then we subtract the mean or the average from each one of those numbers. Again, remember this is all it's all in its own little parentheses here, so we'll subtract. So like if we use the same numbers, 3, 4, and 5 as our numbers that we had in our set. So we'll do 3 minus the average, which I think the average is 4 because 3, 4, and 5, 7 plus 5, that's 12. 12 divided by 3 is 4. Yeah, so 4 is the average. So 3 minus 4 squared. And then we add that to 4 minus 4 squared. And then we add that to 5 minus 4 squared. So if you see here, I did that as an example. So when you figure that up, it's 3 minus 4 is negative 1. Negative 1 times negative 1 is still just 1. 0 times anything is 0, and then 1 times 1 is squared. Remember, if you square something, it's not going to be ever negative unless you're using some kind of imaginary numbers that we definitely don't do in biology. So that becomes your numerator, and then you divide that by however many numbers you have minus 1. So in this case, our denominator is going to be 2 because we have 3 numbers, so 3 minus 1 is 2. So we divide that where we added all that up, so 2 divided by 2. And so that we get 1. So now we're looking at the square root of 1, which is just 1. And you have a calculator for that, so don't let that scare you. I'll make this go away. Go away. All right. So standard error of the mean is a little bit different from standard deviation because standard deviation is saying, here's the average. How much is the data spread out? So if it's super spread out from the average, that means that the average is not really a very good measurement to use to reference that data. So if you've got a whole bunch of super spread out data, and saying the average isn't telling you a whole lot because you've got a whole bunch that are very, very different from that average. Standard error of the mean is trying to figure out how different your sample is from the actual thing that you're trying to sample from. So my example here was a field of sunflowers. So if you have 5 sunflowers and that's your sample and you're trying to find their average height or something, or trying to find their height. So you sample 20 sunflowers out of, I don't know, thousands of sunflowers that are in the field. So obviously you can't count every single one, so you take a sample. But the bigger the sample, the better, because what happens here is you do your standard deviation just like you have in this part right here. So you find how spread out it is from the mean, and then you divide that by the square root of however many you have. So the square root of, well, for math purposes, we'll say 25. The square root of 25 is 5. So if you had 25 sunflowers, you'd be dividing that by 5. If you had 2,500 sunflowers, then you would be dividing that by 50. So the bigger the n is, the bigger your denominator is, so the smaller your standard error is. So this is the whole thing that explains why a bigger sample size is better, is this calculation right here. So the bigger your sample size, the bigger your denominator is, so the smaller your standard error ends up being. So it's just a measurement that is, it's a statistic, it is a statistical analysis of how good your sample is based on how big the thing you're trying to estimate is. So if you're trying to estimate the average sunflower height in a whole giant field, the more you have, the better, because it's just a more accurate measurement because, and this explains it, I guess. So that's my best explanation of that. Chi-squared is something that we are going to use when we get to genetics a lot. This is a terrible sounding thing, but it really, really, really is not that bad. So what the purpose of this is let's say that we think that something's going to be like 50-50. So you get half this and half that. But in reality, you see 30% of one thing and 70% of the other. So what this calculation does is helps us figure out, is that actually different, or could that difference be just due to random sampling differences? So it's saying like, okay, is there an actual difference between what we expected and what we observed, or is what we observed just different because something random just caused it to be a little different? So it's giving you an actual statistical measurement of that. So again, we have our sum, right? So for each individual observation, we would do this little fraction here, and then we would add all of that together, and that gives us that x squared, the chi-squared value. That is the Greek letter chi. So the observed value is what you actually measure. The expected value is what you expected. So that was where we said like we expected it to be 50-50. So if we had 30% of one, so that'd be 30 minus 50, and then we'd square that, and then divide it by 50. And then we'd do the same thing for 70 minus 50, square it, divide it by 50, and then we would add that up. And that gives you that chi-squared value. Or if we expected everything, or expected 75% dominant and 25% recessive of something, which I think is the example. Oh, I'll get to that. I have that example on the next slide here. So once you find that chi-squared value, then you have to use that scary looking table where you have to compare it to these values here. And basically what this is, you're trying to say, I either accept or reject my null hypothesis. So null means none or nothing. So that's saying there's no difference, saying that if that what we saw is a little bit different than what we expected, but not different enough to actually say it's legitimately different. Whereas the alternative hypothesis is saying that there is something different that's causing it to have different values. So this table right here, generally you'll be given this p-value just means like how certain are you that it is not due to random chance, that is totally different, which is very confusing. It basically you're usually given this, so they'll say use a p-value of 0.05 or something like that. So the degrees of freedom is however many numbers you have minus 1. So basically it's like how much wiggle room do you have? Because obviously if you had 50 different numbers that you're trying to, compare the observed and the expected, when you add all that up, it's going to be a bigger number. So you notice as you have more degrees of freedom, that number you're comparing things to gets bigger and bigger and bigger. So just because naturally if you have more numbers, it's going to be a bigger thing that you're dealing with there. So in our example of like the 50-30 or whatever, that means we have two different measurements. So we would go back and it's 1 degree of freedom. So we're looking at these numbers here. So let's say with p-value of 0.05, and when we add that up, if the sum of all of that stuff up here, when we do the 30 minus 50 squared divided by 50, and then the 70 minus 50 squared divided by 50, if that added up is less than this number here, where the p-value of 0.05 and our degrees of freedom cross, then that means that there was not a significant difference between them. So that makes sense. If it's a smaller number, that means that they were closer together, so there's not much of a difference. However, if it's bigger than that number, that means that there was a bigger difference between the numbers, and therefore there is a significant difference. So something legitimately caused that to be different versus just just random sampling differences. So an example here, you'll usually be given a chart like this or something. So this is the best way to figure this out. So with your chi-squared, remember you have to do the O minus E squared divided by E and then add it all up. So it breaks that down for you. So let's say we have, like that very generic Punnett square where you have two heterozygotes, and you end up with 3 out of 4 that are dominant and 1 out of 4 that's recessive. So that's what we're saying we expect to see for these little bunnies here. So we expect to see 75% of the dominant trait and 25% of the recessive trait, because we are both heterozygous. But when you perform it, you see 70% dominant and 30% recessive. So we're trying to figure out if something else was at play there, or if that was just random whatever with just random things that are not actually significant. So when we do this, so we expected to see 75, 25. We observed 70 and 30. So then we do the O minus E for each one, and then we square each one of them. We divide each by what they were expected to be. So 75, 25, and then we add those numbers up. So we do that individually for each one. So we subtract, we square it, we divide it by the expected, and then we add all that up. So our value here is 1.33. So we have two groups, which means our degrees of freedom is 1. So we always take 1 away, so that gives us our wiggle room, right? And if the p-value is 0.05 and our number was 1.33, so 0.05 and 1 degree of freedom, 1.33 is less than 3.84. So therefore, that's not statistically different than 75-25. It's not nearly as crazy as what it looks like or sounds like. You are literally just saying, okay, here's a hypothesis that says nothing happened. There's nothing different. That because these numbers are different than this one, it literally means nothing. And the other hypothesis says that something else affected it and actually legitimately caused it to be different. We figure out what the results were supposed to be, so that's kind of like your control. And then we found out what they actually ended up being, so that's what we saw in the experimental values. And then we did this big old fancy calculation that you don't have to understand why it is the way it is. You just have to know how to do it. And that gives us the relationship between that expected and observed value. So it says, okay, here's how different it actually is. Here's how different what we actually saw is from what we thought it was going to be. So then we look in the chart and compare it to a number based on the strength of the probability. So 0.05 or 0.01, you generally will be told what that is, although you can assume it's 0.05 if you're not told, because it almost is always 0.05. And then how many groups we had, because obviously the number that you get is going to be bigger if you have more groups because you're adding stuff up. All right. If your fancy calculation was less than that number, so less than whatever number was in that chart, then you accept the null because it was small enough that nothing was weird. It was close enough. And if it was bigger than that number, we reject that null because something must have actually happened. So we're just trying to figure out if what we saw was actually different than what we thought we were going to see. It's just a number that says whether or not what you saw is actually different than what you thought you were going to see. Okay? So the bigger that number, that bigger the difference, because that totally makes sense. And so if it's too big, then that means that there is actual differences happening. And if it's not very big, that means that it was probably just something random. Okay. And then over here if you look over here at the thing on this side, so that all that was right here. And then to the right of it over here, it gives you a key for some of those variables, so you don't have to memorize exactly what they are. And I told you what all these things were, but it gives you that key there right on the equation sheet. All right, laws of probability. These are way easier because they make sense. So mutually exclusive is the only thing you probably don't know what that is. That means that things are not happening at the same time. They cannot happen at the same time. So we're looking at things being things or, because you can't. So what it's saying is that you can't have A and B happening at the same time. So the chance of this or that happening, you add it together. So for example, if you are flipping a coin, the chance of getting heads is 50%, and the chance of getting tails is 50%. So the chance that you get heads or tails is 100% because those are your only two choices. If you roll a dice, the chance that you get a 1 is 1 out of 6. The chance that you get a 5 is 1 out of 6. So the chance that you get a 1 or a 5 is 2 out of 6 or 1/3. So those things can't happen at the same time. You can't roll both a 1 and a 5 unless you have two different dice. So that's what that is. So if you ever see the word or, that's what that's talking about. And then if independent values means that they can happen at the same time if they want to. So I'm an independent woman. I can do what I want. So they can happen at the same time. So if they do, you use the word and. So that is where we have two coins or two dice. And so we say, what is the chance that this coin is heads and the second coin is heads? So in that case, that's a much lower probability, right? Because you have 50% chance that this one on in your right hand is going to be heads, and then you have a 50% chance that the one in your left hand is going to be heads. So in this case, because you have to get this and that, you multiply those probabilities. So 0.5 times 0.5, so 50% probability times 50% probability gives you a 25% probability, so 0.25. So you get a smaller probability because they both have to happen. Or if you have a dice, so you have two dice, so you're playing Monopoly and you're rolling two dice, and so everyone wants to get 12, so everyone wants 2 6s. So the chance that you get a 6 on both dies, you have to have one out of 6 chance on one of the dies and 1 out of 6 chance on the other one. So the chance that you get 2 6s at the same time, a 6 and a 6, is 1/6 times 1/6. So it'd be 1 out of 36. So if you ever get that, that's pretty lucky. And we see that here in the Punnett square. You have been doing this without realizing it. So if it's a perfectly half chance that you get a big A from mom versus a little a from mom, and a perfectly half chance that you get a big A from dad and a little a from dad, so then the chance of getting 2 big A's is 1 out of 4 when you fill in the Punnett square. That's literally all a Punnett square is these laws right here at play. So the chance of getting a big A and a big A is 50% times 50%, or 1/2 times 1/2, which gives you 1/4. So you only have 1 out of those 4 boxes that has both of those because it had to get both. What is the chance that you get a big A and a little A? So chance that you get a big A from dad and a little a from mom is, so big A here and little a here is 1/2 times 1/2. So that's 1/4 chance that you get that combination. Or you could get a big A from mom and a little a from dad. So it's 1/4 chance that that happens. It's 1/4 chance that that happens. So to be heterozygous, you can be this or this. So you add those together to make 1/2 chance of being heterozygous. So you have been using these, even if you don't realize you have been using this. Hardy-Weinberg is basically the same thing as that, except it's if it's not perfectly 50-50. So see here, like P is the allele 1, Q is allele 2. So you can think of P being like how the frequency of big A, and Q is the frequency of little a. So if it's perfectly 50-50, the chance of getting big A, the 2 P's here, you square the chance. So 1/2 times 1/2 is 1/4. So this is basically a Punnett square, but this is for if, let's say that it changes and you have 90% of the alleles in the population are big A and 10% are little a. So then the chance of getting two big A's is 0.9 times 0.9, which is 0.81. So now you have an 81% chance of getting both. And oh, I have that same example down here. It must be where my brain goes. So then to get two little a's, little a is 0.1. If big A is 0.9, that means a little a has to be 0.1 because those are your only two choices. So that's what this part is for. They have to add up to 100%. Okay, so if big A is 0.9 and little a is 0.1, then to get two little a's, you have to do 0.1 times 0.1. So there's only a 1% chance, 0.01 frequency, so 1% chance that you would be little a, little a. So then to get heterozygous, you do so chance of getting a big P, 0.9, and a little, or big A and a little a, so 0.9 times 0.1. But again, just like here, there's two ways of getting that, so you have to times it by 2. If you're confused, it's okay. We'll come back to it. But this is not different than a Punnett square, except for the fact that you can put something in other than 1/2 and 1/2. So this is if the alleles are not perfectly half. It has the metric prefixes on here. I put this as a guide for you in case you are totally not remembering scientific notation. So 1 times 10 to the 0 is 1 because anything to the 0 power is 1. And then if you're going for the positive powers of 10, it's however many zeros it's got. Like 10 to the 1 has 1 0. Oh, that should be that should be up here. 10 to the 1 has 1 0. 10 to the 2 has 2 zeros, 10 to the 3 has 3, and so on. With the negative ones, it's you move the decimal place over however that many times. So essentially 10 to the negative 1 means 1 over 10 to the 1. So that means 1 over 10, which is 0.1. And then 10 to the negative 2 is 1 over 10 to the 1 over 10 to the 2, which is 1 over 100, so we get 0.01 and so on. A rule of thumb you can use for this is it's going to have the number of zeros here is this number minus 1. So there's 11 zeros here. So there's 5 here where it's -6. There's 2 here where it's -3. So basically you took the decimal place from behind the 1 and moved it over 12 times, or moved it over 9 times, moved it over 6 times, and so on. It also has the names of the different metric prefixes. This is the statistical definitions that you would need to figure stuff out. If it says like find the mode of a set of values or find the median or mean or whatever. Mode is just if you see one number more than once, or the number that you see most often. So if you've got a set of numbers like it's 2, 3, 3, 5, 6, then the mode is 3 because there's more 3s than anything else. There's 2 3s, whereas there's only one of each other thing. Median is if you put them all in order and you take the one that's in the middle. Mean is if you take the average. And then range is figuring out the difference between the biggest and the smallest. So it's trying to figure out how spread out your data is. Statistic stuff is my least favorite thing ever, so that's terrible. Anyway, back is not nearly as bad. And so that was the whole front of the equation sheets. So now we're going to go over this stuff, which is more actual biology ideas. So population growth, this looks terrible. It is not. It totally makes sense. So rate here is talking about, so dy is the amount of change in the number of organisms. And dt is the amount of time that change happens in. So typically, this is like a year or a month or something, the dt. So d means change in time. So dt is change in time. Here is y is the, I don't know why they use y, but it's the change in the amount of, or the number of organisms. So we'll say in a year, there were 10 more tigers than there were the year before. So then dy over dt is 10 tigers per year. That's all that is. So that's if you know exactly how you counted at the beginning of the year and you counted at the end of the year. And so you know exactly what the difference there is. This population growth is the same thing, except you find it a different way. So the B here is the birth rate, which is the number of births. D here is the death rate, which is the number of deaths. So DN, so it means a change in the number of organisms in a year or whatever, is however many were born minus however many died. So if 50 were born and 30 died, that means that it increased, 50 minus 30, it increased by 20 organisms in that time frame. So that's what this right here explains on this page. So then exponential growth, that's where you have like the J curve. So I have here the J curve is exponential, it's because it's shaped like a J. So it starts growing and then it grows faster and faster and faster and faster and faster and faster and faster because there is no limit whatsoever that's keeping it from stopping to grow. So the R max is the maximum growth rate. So that's saying if these bunnies or these bacteria are reproducing as fast as they possibly can, this is the maximum rate at which they can reproduce. So let's say that bunnies, the maximum rate they can reproduce is 0.5. What that means is every individual per year can make 0.5 more individuals. Or if the R max is 1, then that means that every individual makes 1 new individual every year. Or for every individual, you get 1 new individual every year. Or if the R max is 5, that means that for every 1 individual, the next year there will be 5 more. So R max is the fastest it can possibly grow. Typically, it's less than 1, but it can be bigger. So for exponential growth, there's no stopping that growth. So if you're going to get 1 new individual per individual every single year, then the first year you have 1. So then you have, no, I guess we're talking about bacteria. So if you have 1, then the next year you've got 2, but then there's 2 individuals that can make 1 new individual each. And then the next year you have 4. And then the next year you have 8, and then 16, and then 32, and then 64, and then my math is not working anymore after that point. But it starts increasing like super fast. So you're just multiplying that rate times however many you had when you started, and that's the increase or decrease, actually. It could also be negative, but typically we look at it as positive. That's however many individuals it increased by that year. So if you don't know how many births and deaths there were, then you can figure it out based on the typical R max of that population and the number of organisms there are in that population. So this is the population growth one here is kind of like after the fact you can count. Exponential growth is a prediction without a limit. Now, logistic growth here, notice it's the same thing, but you add this little thing right here. K is the carrying capacity. So it has to stop growing at some point for these populations because there is some kind of limit, whether it's not enough shelter or food or something. And so once it reaches that, or once it starts getting close to that, see it starts slowing down, and then eventually it will stop. So the reason why is this little K minus N over K thing here. So if you look at this, K minus N over K, if K is the carrying capacity and N is the number of organisms, it makes sense that growth slows down when the number of organisms starts getting close to that K level. So let's say that K is 100 and N is 5. So then 100 minus 5 is 95. So 95 divided by 100 is basically still just 1. So it's not really going to be different than what the exponential growth is because you're taking whatever you got for the exponential growth and timesing it by 0.95. So it's a little bit slower, but not a lot slower. But if there were 95 individuals and a carrying capacity of 100, that means you do 100 minus 95, so that's 5 divided by 100. So now you're taking what could potentially happen with exponential growth and timesing it by 0.05, which is going to be significantly smaller than what you get for exponential growth. So that is something that once if n is equal to k, so like if n is 100, so 100 minus 100 is 0. So 0 divided by 100 is 0. So that means there is no change in organisms. If it's at the carrying capacity, it will stay at the carrying capacity. But the bigger the N is, the lower this gets. And this actually can be negative. So if K is 100 and N is 150, that means that that's going to be negative 50. 100 minus 150 is negative 50 divided by 100. So then this right here is going to be negative 0.5. So then you'll take what you got for exponential, times it by negative 0.5, and your population growth is going to be a negative number, which means that the population is going to go down. So here I showed you if it does happen to shoot above the carrying capacity, because organisms don't look at this equation and say, okay, well, we can only make this many. So they just reproduce, and then if there's too many, then more die. So if it overshoots, then N is going to be higher than K, so then that value is going to be negative, so then the population goes down. But then it dips maybe a little bit below K, so then the population is then the growth rate can be positive because N's a little bit less than K, so then it can go back up. But then, oops, it overshot again, and then it kind of equilibrates there. So exponential growth is the fastest it can possibly grow, and then logistic growth is the fastest it can possibly grow, but it's limited by how close you are to the carrying capacity. And this is a plug and chug kind of deal. So as long as you understand what these different variables mean, then you should just be able to plug it into the equation and just run with it. Just be aware that the Rmax is the maximum growth rate, the fastest that it can possibly grow. And in logistic growth, it will never be that fast unless N is 0. Okay. Simpson's diversity index is actually easy, and it makes sense too. So diversity is looking at, like, biodiversity is looking at the number of species in an area. So higher biodiversity is a good thing. So invasive species and pollution and coal burning and all this kind of stuff decreases biodiversity, which is a bad thing. So we're looking at diversity index is helping us to kind of measure how much biodiversity there is in a certain area. So why this is set up the way it is, you don't have to know. But what it's telling you is how biodiverse is this particular community. So let's say, so my example here, and we'll talk through this. So we're looking at a dirt community. We're trying to figure out how biodiverse the dirt community is. So if we have 100 worms, 20 beetles, and 180 roly-polies, and it gives us 300 total organisms. So then we have our little sum thing there again, remember? So we have to do this part right here for all 3 of those numbers, and then we add them all up, and then we do 1 minus whatever that is. Okay, so we do 100 divided by the total, 100, so for the worms, times it, and we times it by itself, so we square it, and then we do 20 divided by 300, and then we square that. And then we do 180 divided by 300, and then we square that. So I showed the walking through the calculation here, but basically, so this outside parentheses right here is basically that. It's basically that epsilon saying that you're adding it all up before you do anything else. If you add it all up, 1 minus that is 0.526. Now, if there were 5 different species in the area and they were all equal, then the diversity index would probably be higher. But I'm going to give you a practice problem for that. That's what I'm going to do for that. You'll have a practice problem. But it's easy to plug it in as long as you know how to use it. So whatever comes after that sum happens individually for each number, and then you add all those up. Okay. Water potential is explaining osmosis. This is a terrible looking thing, but it totally makes sense. Okay. It's explaining why water moves a certain direction. So it explains why water moves from where there's more water to less water. Or why, whenever something is hot, it spreads out faster. So it's kind of, or if you put pressure on something, then that's why the water is going to squirt out of it. Okay, so this is explaining that perfectly. So the pressure potential, which is if you're pushing on it, it's pressure potential. The higher the pressure, the more the water's going to want to leave whatever's got pressure on it. Solute potential is looking at, like, how much solute is in it. So remember, we've done this with, like, a red blood cell in a beaker. So if the red blood cell is in a like super salty water, the super salty water is going to pull water out of the red blood cell. Okay, so the solute potential is going to be based on the amount of salt or sugar or whatever that's in the water. If the thing is in an open container, the pressure potential is 0 because nothing is pushing on it. Okay, so we're assuming that everything's neutral at just the regular atmospheric pressure. Okay, so if it's in an open container, that means it's not like in a vacuum pressure cooker thingy that's pushing on it and causing stuff to come out. All right, to figure out the solute potential, so we're figuring out like how much water things want to give away. So the more positive this value is, the more water it has to give away. Ionization constant here is the first thing. If it breaks up in water. So in chemistry, you learned that ionic things, like for example, salt is NaCl. So when you put it in water, it separates into Na plus and Cl minus. So instead of just putting one molecule in the water, you actually put two things in the water. So it's separated into two. Whereas sugar, when you throw a sugar molecule into water, it just stays a sugar molecule. It's just one thing. So it's not ionic. So if you have an ionic compound, then it's going to separate into however many ions it's got. So if you've got like, I don't know, carbonic acid or something, then it's H2CO3. So that means you have two H pluses and one CO3 2 minus. I'm sure you remember that from chemistry, right? So it's however many pieces it breaks up to whenever it goes into water. So how many pieces it ionizes to when it gets in water. I'm willing to bet you're probably not going to see anything in an example for this other than like sugar or just regular salt or something. They would tell you what ion it breaks up into probably. Or it would be very, very obvious. But anyway, so however many pieces it breaks up into, that's what you put here. So sucrose or sugar, any kind of sugar is going to be 1 because it does not ionize, whereas salt, this value would be 2. But the reason they put sucrose on here is because generally that's the example that they use. Okay. The molar concentration is going to be how many moles per liter that are in there. And this is one of the reasons why having chemistry first is very nice, because you have to do concentration when you're in there. So molarity is a measure of concentration. Moles is really hard to explain. So if you haven't done that for a while, I suggest looking back at your chemistry notes or looking at a Khan Academy video or something talking about how to find molarity. So essentially the concentration. So a lot of times you'll just be given the concentration. So if they say the concentration is 1 molar versus 5 molar versus 0.3 molar or whatever, then you can just plug that in into the equation. And I'm going to give you some practice, and it will have this molar in it. So you won't have to do any kind of fancy stuff to it. And then the pressure constant is going to be this number. I doubt that because this is not super advanced mathy stuff, they're probably not going to make you convert things to this unless they say like, they're probably not going to make you convert that. That's probably the number you would use. Then just plug that in there. So you have the ionization constant, the concentration, the pressure constant, and then hidden down here under my toolbar, because my toolbar won't go away, is T, which is temperature, which needs to be in Kelvin, not Celsius. So if you have a temperature in Celsius, you need to add 273 to it. And on the actual equation sheet, it does say that, but for some reason on my slide there, it's hidden because it doesn't like me. So if you see here, it does have degrees Celsius plus 273. So if you're given that, then you just have to, if you're given that, you just have to convert it, or you can just plug it in, let's see, did I have I did have an example, sweet. So if you were given 0 degrees Celsius, then you would just add 273 to it inside the equation, and you just plug that whole thing into your calculator altogether. So if you have sucrose at a 0.5 molar concentration, this is your pressure constant that it gives you there. And then you had 0 degrees Celsius, so you add 273, you multiply it all together, you get negative 11.3. So that is your solute potential for that. Oh, so that is your solute potential for that. And so if you have, so that what that means is that if you have something that's got a more positive water potential, then anything with a more positive water potential is going to push water to anything that is less positive. So it makes sense if you think about it like if the water potential of something is 3, and then the water potential of the other thing is -2. So 3 is going to push to -2. That makes sense. But you just need to make sure you're looking at this really carefully because most of these values are going to be negative. Because if you look at this, the solute potential is required to be negative. Ionization constant cannot be negative. Concentration cannot be negative. Pressure constant cannot be negative. And your temperature, because you are using Kelvin, cannot be negative because 0 Kelvin is as cold as it can get. Okay, so the solute potential cannot be positive. The highest it can be is 0. So if you are in an open container, then your water potential is going to be negative because your pressure potential is 0 and your solute potential has to be negative or 0. So, I mean, unless your water potential is 0, then it's going to be negative. Okay, so just make sure you're looking at that really carefully. So, this explains why why soil pushes water into the roots and roots pushes water up the trunk into the leaves. Okay, so the water potential of the roots maybe, so the soil maybe has -3, maybe the roots have -10, the leaves are -20. So -3 is more positive than -10, so it moves toward the -10 in the roots. And -20 is less than -10, so it'll move from -10 in the roots up into the -20 at the leaves. So that explains why water moves from the soil to the roots all the way to the leaves. Because the water potential is consistently less and less and less, more negative. So don't look at that and think, oh, it's bigger if it's -20. It's not. Make sure that you look very carefully at that because it is very, very easy to look at that wrong. It also explains why water would leave a red blood cell if it's in a super salty water, because if it's in super salty water, that means that this concentration is really high, which makes this number a really big number. So it's negative. It's a big negative number. So it's going to be super negative for the salt solution because the concentration is so high. So it will probably be a more negative number than the red blood cell. So if the salt, they'll say that the concentration of the salt in the water is like 3 times as high as the concentration of the salt in the cell. So then this would be 3 times as negative. So it would definitely move toward the more negative. So that's why it moves out of the cell. Okay. pH is also something you're not going to have to do a whole lot with it. But what pH is a measure of the amount of hydrogen ions that is in the water. So the more hydrogen ions that are in the water, the more acidic it is. The less hydrogen ions that are in the water, the less acidic, which means more basic it is. Okay, so this right here gives you a scale showing you what the concentration of hydrogen ions are as compared to the numbers that go with the pH. And notice that it also includes hydroxide. So H plus is your acid molecule. OH minus is your basic molecule. So notice that they're backwards here. Also notice that this has an exponent essentially of 0, right? 10 to the 0 is 1. This has an exponent of -14. That also adds up to 14. That adds up to 14. That adds up to 14, and so on and so forth. So they're essentially like inverses of each other. So if you have more hydrogen ions, you have less hydroxide ions. Because remember, this is a -14 there. So that's a really, really, really tiny concentration of hydroxide whenever it's the pH 0. You just need to know how to put this into your calculator, but also you may just get lucky and they give you something super simple. So notice here, we'll go with the hydrogen concentration of 10 to the -3. That gives you pH 3. You literally just take the number that's in the exponent and stick it up there. That's it. It's just not negative. Okay. If they give you the hydroxide concentration of 10 to the negative 4, that knows, so 10 to negative 4, that means that hydrogen has to be 10 to the negative 10 because 4 plus 10 is 14, right? So that means the pH is going to be 10. So that actually, negative 4 is right there. So negative 4, negative 10 gives you 10. If it gives you something crazy like 9.8 times 10 to the negative 7.3, which it very possibly could, you would plug it into your calculator like negative and then hit the log button and put that concentration in there, and then it'll give you the pH. So this is going to be something probably around like, but it's going to be somewhere between 7 and 8. We know that because this is negative 7.3. So there's easy ways to calculate that if they give you easy numbers, but then if they give you something crazy, your calculator will very easily do that for you. And on the AP exam, you can either have a scientific calculator or a graphing calculator, and I believe you can bring your own, assuming they test you in person. And then the last part here shows you surface area versus and volume, so it just gives you the formulas for how to figure that out. I'm going to trust that your math teachers have done a relatively good job of teaching you how to use these equations. I certainly hope so. But the reason why this is in there is because if the surface area is high versus the volume, so we take the ratio. So say the surface area is 20 and the volume is 1, so that's a 20 to 1 ratio versus maybe a 1-to-1 ratio. So let's say that the surface area is 10 and so is the volume. So the higher the surface area versus volume, that essentially means that it's got like a really, really, really big outside. Like, what's a good example of that? Okay, piece of paper. A piece of paper versus a ball of some sort. So a piece of paper has a very, very small volume, but the surface area is humongous. Whereas the ball has a lot bigger volume as compared to the surface area. Your skin cells, your skin cell versus your white blood cells is exactly that same comparison. So your skin cells are nice and flat, like a piece of paper, and your white blood cells are basically spherical. So the point of that is the higher the surface area to volume ratio, the better and easier it is for a cell to get materials in and out. So imagine trying to get something from the center of a piece of paper to the outside. It's not very far to travel. You got lots of different ways it can go, and it's super easy to get out. Whereas, well, I guess assuming there was an inside to a piece of paper, but whatever. Versus something in the very center of a ball trying to get to the outside. So relative to each other, it's a lot easier for the piece of paper than it is for the ball. So that's the point of that. So chloroplasts and mitochondria, chloroplasts have all those, the thylakoids that are stacked up, little discs, individual discs, and they're nice and flat. And then the mitochondria has all those ridiculous folds. And the reason why it has so much of the membranes in there is because it's trying to increase its surface area versus its volume so that it can get stuff across those membranes, because we'll find out later that for chloroplasts and mitochondria and photosynthesis and cellular respiration, respectively, require moving things across those inner membranes. It's very, very, very important for that to happen. So they adapted to whatever environment they needed to do that in, and they and as they adapted, the ones that had higher surface area did things more efficiently, and then that's what came of it. So that's it. I'm going to give you some practice for it, but this is an explanation there. You will also always have access to this PowerPoint if you ever need to reference back to it. There are a couple of different examples on here that I didn't go over, but kind of toward the end, I think I started using the examples that I had on the slides. I apologize for the technical issues in trying to get the slideshow running versus the slides here and then things getting covered up and all that, whatever. But you do have access to that all the time. Hopefully I was not too confusing. If you have questions, you can ask me.